Okay, could somebody confirm that you can hear me?
Yes.
I can hear you.
Thank you. I tried to log in with Zoom, meaning going to www.zoom, and apparently Clark College has changed my password, so I wasn't able to get in. So I have to come in through my class, and it doesn't have a background on the screen. Let's see. All right, are there any questions?
I had a question about the extra credit work
Okay,
log,
the worksheet?
the study sheet.
Yes.
So from my understanding is we write a plan on the first sheet, right? The schedule.
No, you put your schedule on the first sheet. You then,
Right.
on the second, what do they call that, tab, you put your study schedule for week 1.
Is
If you,
that counting the other classes or just this one?
just this one.
Okay, so for log 1, it would just be microbiology.
For the first tab, it's going to be your daily schedule. On the second tab, it will be the times you're planning to study for microbiology.
Okay. Do you mind if I screen share at the end of the Zoom?
That's fine.
Okay.
However, you're going to have to do it during the lab. There will only be 10 or 15 minutes between the lecture and the lab. And so I can't do it in that period. Okay. So it have to be after we get started in the lab.
Okay, that's fine.
Okay.
I had a question too. So if my schedule's the same always, then I just do the first one, right?
You do your daily schedule
Yeah.
in the
But
first, and then in the second one, you do the study schedule. And if all the weeks are the same, you only do one.
Yeah.
However, if you have different study schedules on different weeks, put those in as different tabs.
Okay. Yeah, I just did the one with everything.
Okay.
Because it's the same.
All right, let me go ahead and erase this. You can't see this, I know yet, but
And for
this is where we
questions,
left off, huh? Go on, another question?
yeah, so for the lab 01, if we have questions, can you answer it in the next class?
In the lab, I will.
And then are these recorded?
The lecture and the lab are normally recorded. They will be recorded unless there is a unforeseeable problem. The first lesson, lecture, and the lab have been recorded and uploaded to YouTube.
Okay, and to get to your YouTube, we just go on Canvas, right?
You can go there directly. You just need to know the site, or there's a link in my Canvas webpage to my YouTube site.
Okay, thank you.
All right. All right. Let me get ready for class. I think I'll leave that for the lab. Let me close that. And I need to get the syllabus up. Sorry, I normally do that while people are asking questions, but today I got distracted. All right, let's go. Is that the right term? Yes. No. That's winter 21. This is winter 22, 23. I just want to show you the schedule. So, all right, let me share my screen now. You should be seeing the screen. Boy, there's a bunch of messages that are starting to annoy me. A participant has enabled closed captioning. I assume that was me. All right, so today we're going to hopefully continue chapter 1 and get well done on it, maybe start chapter 10. And there is a lab today in the lab session, which is, I think, at 6:30, and that'll be covering lab 1. Any questions about what we're doing? Please note that next week there will be a practice quiz, and it's called Practice Quiz 00. There will be 2 of these. Let me go home. There will be one that'll be having multiple choice questions under week 2 lecture. There it is. And that will be the practice quiz 00B. It will require that you use Respondus Lockdown Browser. So you need to download that and get it running on your system. If you do not get it running on your system, then you should contact the computer help desk. And you should do that before the quiz is due. Now, this is a practice quiz. It'll be worth a few points of extra credit. I don't remember how much it's worth, so let me look into that. Practice quiz 00A does not require Respondus Lockdown Browser. It will be fill in the blank answer questions. It says it's worth 0 points, but each of the quizzes will be worth 1 point of extra credit. And that's why it's worth 0 points, because you'll get like up to 1 point for each quiz out of 0, meaning 1 point of extra credit. Any question about any of that? The point is to help you become familiar with my quizzes and my quiz questions. And so this is an extra credit quiz. You should take it just to make sure you get Respondus Lockdown Browser running on your system. Because last time I had 2 students who waited to the very end. One of them didn't have any trouble getting Respondus Lockdown Browser to run, but the other student didn't get it running. And then she contacted me. I contacted her to tell her to get the computer help desk. And of course, by the time she got it running on her system, the answers were showing, so she could not take the quiz 1. So get it running on your system before the time expires to take the quiz. And if you take the practice quiz, that should do it. Any questions about any of that? All right. Let me remind you, there is 2 extra credit assignments, which I think are due this Saturday. There is the extra credit study sheet, and then you can submit a picture to me of your face for extra credit also. I think the picture is worth 3 or 4 points, and the study sheet is worth something like 6 points. Next week, we'll talk about another extra credit assignment. Oh, and then one other thing, you need to take the plagiarism tutorial and the plagiarism quiz by this Saturday. You must get 100% on the plagiarism quiz, or you'll get 0 on the infectious disease project. And that's 100 points you're missing. Any question about any of that? If not, let's continue with the lesson. we were talking about where you can find microbes, and essentially is everywhere on the planet, every inch, except where there is lava or magma or something that's really hot. So microorganisms are found on the land and in the water, in the atmosphere, subsurface on both under the land and under the ocean, on plants and animals. Essentially, wherever liquid water is present, you can find life. And then living things can be found between temperatures of about a minus 20 degrees Celsius and 120 degrees Celsius. Remember, boiling water is at 100 degrees Celsius and freezing water is at 0 degrees Celsius. So that temperature where we find life is a minus 4 degrees Fahrenheit and 250 degrees Fahrenheit. The things that are living at a minus 20 degrees Celsius have their own internal antifreeze so that they are able to be metabolically active at that temperature. Below negative 120 degrees Celsius, something may be alive, but they'll be frozen and they're not going to be metabolically active if they are frozen. Any question about that? And then we can find life in the deep sea vents. And that's why living things can be up to 120 degrees Celsius. All right. Microbes are numerically the most abundant life form on the planet Earth. How many microorganisms are present? And we're talking about prokaryotic cells. We're not talking about viruses. For example, if we included viruses in the microbes, then the numbers would be 10 times the number that I'm going to give you here. But in 1 gram of soil, there are about 10 to the 8th prokaryotic cells, most of them being bacteria and archaea. But there are some microbes which are eukaryotes as well. The numbers depend exactly on how much moisture is in the soil and how much organic matter is in the soil. The more organic matter and well, I can't say the more moisture, but with lots of moisture, you get higher numbers. And then with less moisture and with less organic matter, you get less microbes. Like in the desert, you don't get these numbers in the desert. Any question about any of that? So your average soil in like Western Washington or Oregon would be around these numbers. Maybe even higher because our soil is so wet. Now, do you find more microbes in 1 gram of ocean water? 1 gram of ocean water is 1 milliliter of ocean water. Anyone take a guess? Nobody wants to guess? Well, the open oceans are actually a desert on the planet Earth. So you find fewer microbes in the ocean water. Sorry, I'm hearing a horn going off, and I imagine you don't hear that because it's pretty weak, but it was distracting me. How about in 1 gram of human feces? Do you find more or less than you find in 1 gram of soil?
Would there be more because it's more nutrient dense?
Yeah, there are generally more. And then on 1 human being, more or less. That should be easy, people. Because this is only 1 gram of soil. 1 human.
Oh yeah, tons.
Yeah, you have more. And then the entire Earth, you have a lot more. And let's give some numbers here for these numbers. So for 1 gram of soil, it's around 10 to the 8th, and that's 100 billion. Is that right? Ocean water around 1 times 10 to the 6. Like I said, the ocean is a desert where you find life. And then 1 gram of human feces, it can get up to 1 times 10 to the 11th microbes per gram of human feces. And that will be 55% of the weight of the feces, meaning that's over half the weight. Now, that would vary on how dry the feces are. Like somebody goes to the bathroom in the desert and then you take a look at the feces after it's dried out. It's going to be much less than that. And the low number there is about 10 to the 6. Okay, so that's not coming out of a person. That's after it's dried out. On 1 human, you find 3.8 times 10 to the 13th microscopic cells. That's a staggering number. And then on the entire Earth, you have 1 nonillion prokaryotic cells. That's 1 times 10 to the 30th, meaning 1 with 30 zeros after it. And I bet you didn't know that was a nonillion, but that's actually a name, a nonillion. For comparison, the human population is about 7 to 8 billion. I think we're still a little less than 8 billion, but we're getting close on 8 billion now. Any questions? All right, let's talk about microorganisms and disease. Microbes, if they cause disease, we call them pathogenic. Most microorganisms do not cause disease. Only a small number of microbes are pathogenic. So only a small number of microbes are disease causing. We can break disease into infectious diseases and inherited diseases. Infectious diseases are diseases we will study in this class. And why might that be? Why do we only study infectious diseases? Anyone have a guess?
Because of the bacteria?
Yeah, because they're caused by microorganisms or a microbe. Inherited diseases are not caused by microbes, and so we're not really going to study them. These are diseases that a child would be born with. Who can give me an example of an infectious disease?
Strep throat or Streptococcus A.
Yeah, Streptococcus pyogenes, also called Streptococcus. What is that called? Group A or something like that. And what's another example of an inherited disease?
Alzheimer's.
Alzheimer's. That's not entirely certain that that's all inherited. There might be an inherited link, like Alzheimer's might run in certain families, but Alzheimer's is a little more complicated of a disease than an inherited disease. Meaning,
Would like HIV be one? Because it can be passed to the fetus.
well, HIV is an infectious disease. You get it from the virus HIV. And so if you don't get the HIV virus, you will not get AIDS, for example. AIDS is the disease. HIV is the disease-causing organism, meaning the microbe.
Could Down syndrome count as a inherited disease?
Yes, Down syndrome is an inherited disease. The child is born with Down syndrome. And how the child gets that is they have an extra chromosome 23. that they inherited. So that's a very good example of an inherited disease. Another inherited, sorry, I'm having a hard time saying that word, inherited disease that I imagine you studied in Biology 160 was sickle cell anemia. A child is born with that disease, and they have to have 2 genes for sickle cell anemia. And if they have those 2 genes, then they will be sickle cell anemic, meaning they'll have the disease. Curiously, if you only have 1 gene, it's mostly recessive. And so they generally, at least in a mild climate, it's recessive and the sickle cell doesn't show. Any questions about the difference between an infectious disease and an inherited disease? All right, let's move on. We talked about microorganisms and we said they're small. How small are they? Let me blow this up if I can. Got to get this out of the way. That didn't help. So microorganisms are small. You have to see them with a microscope. You can't see them with their naked eye. For most bacteria, and this is actually a larger bacteria than normal, they're about 1 micrometer wide by maybe a little longer than 1 micrometer long. This one's just a little longer than 1 micrometer long, just a little bit. So most bacteria are between 1 micrometer and 2 micrometers, also called a micron. And those are the sizes we see under the light microscope. Here's another microbe, and this is, I think, a fungus. And this would be like 1 cell there or 1 cell there. And this you can see is much bigger. This would be 50 micrometers there. There's another microbe that looks to me like a white blood cell. And it might be an amoeba, but I think it's a white blood cell. And this is 10 micrometers long. So this might be 60 to 70 micrometers long. This is Volvox. This is a colonial microbe. Actually, each dot here, come on, mouse. Each dot here is a cell. And this is the parent colony of Volvox, meaning it's composed of multiple cells. And it's made baby colonies inside of it. 1, 2, 3, 4, 5, 6 different baby colonies. The parent will open up and that will release the babies to go elsewhere. And then they'll be their own Volvox, grow into their own Volvox. And then this is another lymphocyte, a white blood cell with HIV, the virus, infecting it. Any question about any of that or how small they are? You should note that microbes come in various shapes and sizes. They play beneficial roles in numerous aspects of the planet. We've already talked about them. They decompose our waste that we flush down the toilets. They decompose organic waste everywhere on the planet. Like many a tree dies in the forest and falls down, microorganisms are important in breaking it down. It is true that there are some fungi, like mushrooms, which are not microbes, which can help break down the tree as well. But a lot of the decomposers are microbes. And they're important for recycling the nutrients in dead things. Microbes are also important producers in the ecosystem. Producers are photosynthetic and chemosynthetic organisms that can make their own sugar from carbon dioxide. I think everybody knows that green plants are photosynthetic, and they are the primary producers on land, although there are microbes, which are microscopic, on land, which are also producers. And the point part is that microbes are a significant number of the producers on the planet. I'm sure when you talk about the full numbers, there's many more of them than there are plants. But then the production of sugar on the planet, microbes play a significant part of the production of sugar. And I don't know if they play the significant part, meaning more than green plants, or if it's less. I just don't know the numbers there. But it's a significant production. And then green plants are a significant production as well. Microbes, we already talked about, are involved in the nitrogen cycle, and nitrogen wouldn't cycle on the planet Earth without the microbes. Microbes are also important in producing things such as industrial chemicals, like the ethanol you're putting into your car almost every time you fill up with gasoline in the wintertime, at least in the wintertime in Oregon and Washington. Alcohol is added. And generally that alcohol comes from corn, although the microbes ferment the corn to make the alcohol. And then some other industrial chemicals that are made by microbes include acetone. We use microbes to make various insecticides, and these are important because these are biological insecticides. Generally, they're friendlier on people and the environment than man-made insecticides. We use microbes to ferment our foods. Like microbes actually make vinegar from wine, and we use microbes to make cheese. As far as I'm aware of, every cheese is made with microbes. And then we use microbes to make leavened bread, as well as beer and wine. We use microbes to make products used in manufacturing, like cellulase. We use microbes to make antibiotics for us, like penicillin G is made by a microbe. Some antibiotics are man-made, but some are made by microbes. I think carboxycillin is a man-made antibiotic, and it's in the same family as penicillin G, which is made by a microbe. And then other products used in medicine are made by microorganisms. For example, today, when a diabetic gives himself insulin, that insulin was made in a microbe and then purified. Any question about any of that? By the way, the insulin, it comes from a human gene that was inserted into the microbe. I think it's usually inserted into bacteria. Microbes are important in both biotechnology and genetic engineering. Biotechnology is the use of microbes and organisms to produce foods and other products for human use. Biotechnology is actually centuries old. Like the fermenting of food is, quote, a biotechnology. We don't think of it usually in those terms, but it is. The breeding of corn so that you get something like 80% corn oil out of a seed of corn, that's using biotechnology. And like I said, this is centuries old biotechnology. Genetic engineering is a new technique of biotechnology we're using genes and inserting them into an organism to produce something. Applications of genetic engineering include the production of a variety of proteins from bacteria and fungi, such as the production of vaccines, let me think if I can think of an example of that. Well, there's one. The Sabin vaccine for polio would be made by microorganisms. And I think that's, yeah, the Sabin vaccine's making the whole microbe, but it makes proteins on the microbe. And then gene therapy is another application of both biotechnology and genetic engineering. Gene therapy is when somebody is born with a defective gene, and then we use gene therapy to correct the person, such as a child born without the ability to make antibodies. You can get the correct gene from a human donor, put that gene into the child's cells, and obviously you take out the child's white blood cells and then insert the correct gene into them, grow them up in vitro, and then put those cells back in the child. That would be an example of gene therapy. And now the child can have cells which can make antibodies. And then another example of genetic engineering is genetically modified bacteria, where we can create something with it. Like they inserted a gene into tomatoes to make the tomato not rot right away. They called it the flavor saver tomato. And I think it helped the tomato ripen and then stay ripe. And so this grocery stores wanted it because it wouldn't spoil on their shelves. However, the flavor saver tomato has pretty much gone away. So I'm assuming that you really have to find a store that still has them because they're not easy to find anymore. And I'm assuming that people didn't want to buy them. All right, any question about any of this? If not, let's talk about microbial ecology. Microbes are important in human welfare. One aspect of that is microbial ecology. Bacteria are important for recycling nitrogen, which we've already talked about on the planet Earth, but they're also important in recycling carbon, various nutrients from dead things, sulfur, and phosphorus. And so when something dies, these elements can be recycled and then used by other plants and animals. And then the nitrogen, the sulfur, and the phosphorus are moving around on the planet from one aspect to another by microbes. Any question about any of that? The sulfur cycle and the phosphorus cycle are similar to the nitrogen cycle. It's just there aren't as many steps, but the point is that phosphorus can move around on the planet and be recycled. It can come out of rocks, and it can leach out of rocks by air or maybe wind, or even microbes can leach it out of the rocks. And then the microbes convert that phosphorus into an organic form. Then when the microbe dies, it's released. And initially it'll be in organic form, meaning some chemical like glucose 6-phosphate, for example. But that would then be broken down by microbes, and they'll generally break it down into phosphate and then recycle it, meaning they'll convert it into an organic form. Any question about any of that? All of these examples of recycling of elements and nutrients on the planet are examples of microbial mediated biochemical cycles that support life on Earth. If the microbes did not make the cycle run, the nutrients would stay in one place on the planet. Like when something were to die in the forest, if the microbes and the fungi did not degrade it, the nutrients would be locked up in the trunk of the tree or whatever it is, the tree that's fallen down on the floor, and it would just stay there, all of those nutrients. So the microbes are important for recycling the nutrients. The first to show that microbes were important in the biochemically mediated cycles on the planet were Winogradsky and Bajering. They were the first to show that the recycling of nutrients occur in the ecosystem by bacteria and other microbes. Any question about any of that? Microbes are important to human welfare and bioremediation. Bioremediation means literally life and to remedy. We use living organisms to deal with environmental problems. For example, when you flush your toilet, we use microbes to degrade the sewage. Well, microbes are also important in degrading pollutants and industrial accidents, like this oil spill. They're spraying microbes on it to help digest the oil. Another example would be mercury. Microbes, they can't get rid of mercury, but they can convert it to a less toxic form. So you can use microbes to degrade or detoxify pollutants, turning them into less harmful substances. Like this oil would be broken down into carbon dioxide and water. Any question about any of that? All right, let's talk a little bit about microbes are used as biological insecticides. Some microbes are pathogenic to insects, and they can be used as alternatives to chemical pesticides. And then they will prevent insect damage to crops, as well as prevent disease transmission. We're talking about mainly crop diseases. An example of a microbe which is toxic to insects is Bacillus thuringiensis. Infections by this bacteria are fatal to many insects, and yet Bacillus thuringiensis is harmless to humans, other animals, and plants. Way back in the 1960s, my father bought some of this bacteria and put it on the apple trees. The trouble was he only did it once, and you actually need to do it about every 2 weeks, which is about the same as you're supposed to do with the insecticides to prevent worms in the apples. So my father did get worms, but if he would have done it every 2 weeks, and you have to do it also after it rains, because the rain will wash the Bacillus thuringiensis off the apples, you can use it as an insecticide. And it does work by preventing the apples from getting wormy. Any question about any of that? All right, if not, let's change topics again. We're going to talk about naming and classifying of microorganisms. The scientific names was started by this individual here. Does anybody know who started naming as well as started the classifying of all organisms?
Linnaeus?
Yes, Carolus Linnaeus or Carl Linnaeus, Carolus Linnaeus. He liked Latin so much that he actually changed his, I think he was Swedish, which was something like Carl or Carly, Linnaei or Linnae, something like that. And he changed it to Carolus Linnaeus, which is just Latin. And any of those are correct. It's not a big deal. I'm not going to ask you who started this, but he started it. And what he did was he gave each organism two names. He gave them their genus name and the specific epithet name. This is not exactly the species name because the species name is the genus as well as the specific epithet. So for example, Homo sapiens is the species name, Homo is the genus, and then sapiens is the specific epithet. Any question about any of that? For some organisms, the specific epithet is repeated, not Homo sapien that I'm aware of, but for example, coli. There's B. coli, meaning genus, whatever the B stands for, I don't remember. And there's E. coli, which is genus Escherichia. So all scientific names have two names. For that reason, we call it a binomial naming system, or the scientific nomenclature, or the binomial nomenclature. Any question about any of that? All right, just to briefly, we'll talk a little bit about classification after a few minutes, but I did want to mention that Carolus Linnaeus also started not only giving species a name, a biname, binomial name, but he also started classifying all organisms into different groups. The naming system was so good, we still use his naming system today. The classification is changed a little bit. He classified everything into animals and plants. And as you probably already know, there are more groups than just animals and plants. Carolus Linnaeus did not know about microscopes. Is that right? Did not know about microbes. And so he didn't have the group of microorganisms. All right, returning to names. Names are either italicized or underlined. For example, when I'm using a computer, I can italicize a scientific name, and so I'll do that. But if I'm writing on the board, I can't really write it italicized. So I'll write the name and then underline it to mean that it's a scientific name. The genus name is always capitalized, and the specific epithet is always in lowercase. The scientific name is Latinized, and it's used worldwide. For example, wherever you are in the world, you can say the word Homo sapien, I guess that's two words, and all scientists will know you're talking about humans. With this one, most people would know you're talking about humans. Okay, but like for example, E. coli, which is actually Escherichia coli, if you use that any place in the world, everybody uses it because that's Latinized and that's its scientific name. The scientific name may be descriptive or it may honor a scientist. Like Homo sapiens is descriptive. Literally, it means wise man that walks on 2 feet or 2 legs, whatever you want to call it. E. coli, on the other hand, is honoring a scientist and is descriptive, meaning E. coli or Escherichia coli is found in the colon, and that's why we call it coli. And then Escherichia is named after Dr. Esther, who was an early microbiologist. Staphylococcus aureus is descriptive. Staphylo means that it grows in a cluster like grapes. Coccus meaning that the grape cluster has round cells or objects. Aureus means it has a golden color because Staphylococcus aureus is a gold color. Let me turn my phone off. It's ringing. And I don't want to deal with a phone call during class. All right, any question about any of that? After the first use of a scientific name, the name can be abbreviated with the first letter of the genus and then the specific epithet. So after using Escherichia coli the first time, you can then abbreviate it E. coli. And E. coli is so well known and so well used that even scientists will frequently just call it E. coli. So this is actually in error, but it is an exception, and that's because it's so well known. Almost everybody calls it E. coli. After using Staphylococcus aureus in a paper the first time, you may then abbreviate it S. aureus for all other uses of the name in the paper. Any question about any of that? All right, so that's the naming. Now let's talk about the classifying of microorganisms. And in a sense, we're going to talk about the classifying of all organisms. We first need to look at the classification of all living things on the planet Earth. So we have all living things, and the first classification underneath all living things would be the 3-domain system. When we actually look at life, we see that it has 3 different types or 3 domains. There is domain bacteria, domain archaea, and domain eukarya. The domains are based on the genetic information within the organism, meaning all bacteria are more similar to another member of bacteria in the sequence of its DNA than to any organism outside this domain. So E. coli would be similar in the sequence of DNA, much more similar to the sequence of DNA than it's similar to E. coli is similar to like Staphylococcus aureus, and that would be much more similar in the sequence of its DNA to a member of archaea, such as a methanogen. I'm trying to remember a specific scientific name. I'm not calling one. Or to Homo sapiens, or any other member of eukarya. such as a slime mold. Any question about any of that? So the domains are defined, that group, and it's based on the genetic similarity of their DNA. This came as a shock to scientists because they assumed the archaea and the bacteria were essentially the same because they're both prokaryotes. But when people started sequencing the DNA, they discovered that the archaea DNA are as different from the DNA of bacteria as they are to the DNA of humans. In fact, if you look at it, the sequence of archaea is actually slightly closer to the sequence of Homo sapien than it is to the sequence of any member of the domain bacteria. The reason for that is initially the archaea and the eukarya had a common ancestor, and they had a common ancestor before the bacteria had a common ancestor with that common ancestor. Did that make sense? However, this distinction that these two groups, the domain archaea and domain eukarya, are slightly closer together, it's a real slight, more similar between archaea and eukarya than, let's say, archaea and bacteria. Any question about any of that? All right, no questions. Let's move on to say that our next grouping would then be the kingdom. The kingdom is not defined, so there is no agreement on what makes a kingdom. Not only is the kingdom not defined, but there's a disagreement as to how many kingdoms there are. And scientists have been disagreeing about the number of kingdoms for over 15 years. So there is no agreement as to how many kingdoms there are. You can use 3 kingdoms, in which case domain bacteria has a kingdom bacteria, domain archaea has a kingdom archaea, domain eukarya has a kingdom eukarya. Although there's not much point in having a kingdom if it's the same as the domain. You can also use the 4-kingdom approach, and that's not one that I favor because it lumps the bacteria and the archaea into the same kingdom, and they're already different domains. You can have the 6-kingdom approach, and that's the approach we will use, where you have 1 kingdom for domain bacteria, so they're the same, the kingdom and the domain. 1 kingdom for the domain archaea, so the domain and the kingdom are the same. And then you have 4 different kingdoms in the domain eukarya, such as the plants, kingdom Plantae, the animals, kingdom Animalia, kingdom Fungi, and kingdom Protista. Any question about any of that? Now, as I stated, the number of kingdoms is not in agreement, how many there are, and you can have more kingdoms than just 4 in the eukarya. Let me blow this up a little bit. Some people argue that each of these branches is a kingdom. So it's more than just fungi and animals and plants. There's the kingdom Ciliates, the kingdom Euglenosa, the kingdom Plasmodial slime molds, etc. You can even break the archaea and the bacteria into different kingdoms. All right. So the point is the kingdoms are not defined, and there are disagreements for what the kingdoms are, even among scientists. We're going to use the 6 kingdom approach just because it's easy to work with. And then the bacteria and the archaea are the same as the domains. And then we only have to divide the eukaryotes into 4 kingdoms: plants, animals, fungi, and then the kingdom Protista. Now you should understand that the kingdom Protista is just a eukaryote that is not a plant, not an animal, and not a fungi. So it was always understood that the kingdom Protista was not monophyletic, meaning they all came from one ancestor. And you can divide, especially the kingdom Protista, although you can, if you get enough kingdoms, you could actually start dividing the plants. Any question about any of that? All right. When we're talking about the 3-domain system, you should realize that the cells have one of 2 types. Bacteria have cell types prokaryotes, meaning they're prokaryotic cells. The archaea have the same cell type, prokaryotic cells. And then the eukarya have different cell types. And we call that the eukaryotes, or the eukaryotic cell type. Any question about any of that? All right, the cell type is the terminology your author uses. I just want you to know, you don't need to know it for a quiz, but I just want you to be familiar with the fact that some people call the cell type not a cell type, but a super kingdom. And so you divide all living things into 2 super kingdoms, a prokaryote and a eukaryote. And then you divide the prokaryotes into 2 domains, domain bacteria and domain archaea. And then the cell type eukaryotes is not divided, at least in the domain. So the super kingdom eukaryotes would be the same as the domain eukaryotes. Any question about any of that? All right, so what part is important? The most important part of these 2 slides is you should know that all living things on the planet Earth come from 3 domains, and that the domains are defined by the sequence of their DNA. So if ever we find a new organism, we can find what domain it goes into just by sequencing that organism's DNA. Any question about any of that? All right, let's talk a little bit about the domain bacteria. We already mentioned their cell types are prokaryotes. We find peptidoglycan in the cell wall of the domain bacteria. The cells multiply mainly by binary fission. That's literally the cell divides into 2, and then each half will become a daughter cell. For energy, bacteria can use an organic chemical source, and that should be familiar to you because that's what humans use, such as sugar or an organic chemical source. We use that for our energy needs. But bacteria can also get their energy needs from an inorganic chemical compound, or they can get their energy needs from photosynthesis or chemosynthesis, meaning that they can take sunlight in photosynthesis and then convert it into the sugar they need. the organisms which are not photosynthetic, and they get their energy needs from electrons in another element, like hydrogen sulfide gas. They take that electron and then use it for its energy needs, meaning the cell's energy needs, such as to make ATP. But the point is that we use an energy source for the chemosynthetic organism. And that might be something like iron or hydrogen sulfide gas. And then we convert that into another form, which generally means one less oxygen, electron, sorry, in the atom. You don't need to know that, but that's just what chemosynthesis is. The point is that domain bacteria can get their energy needs from any energy source. Most of the domain members are either free living decomposers, meaning they decompose something that dies, or they are primary producers, meaning they make their own food in the ecosystem. An example of that would be blue-green algae, cyanobacteria. I think we talked about cyanobacteria. Was that yesterday? Not yesterday, last time. Some of the domain bacteria are parasitic, meaning they live on and off of their host. And then some of the domain bacteria are pathogenic, meaning they are capable of causing disease. Any question about any of that? All right, let me briefly mention once again, peptidoglycan is found in the cell wall of domain bacteria, and it's not found generally in other places of the cell. So if you don't have a cell wall, you won't have peptidoglycan. Having trouble moving to the next slide. All right, let's talk briefly about the domain archaea. Their cell type is prokaryotic cells. Does anyone know, do the archaea have peptidoglycan in their cell walls? I'll take that as a no, no one knows. The domain archaea do not generally have peptidoglycan in their cell wall or anyplace else. So peptidoglycan is generally not found in domain archaea. Peptidoglycan is also not found in domain eukarya. So only the domain bacteria have peptidoglycan. Some of the archaea are found in extreme environments, such as some of the methanogens are archaea. They produce methane gas. Some of them are extreme halophiles, like the bacteria living in the Dead Sea between Jordan and Palestine. Most of those are archaea, and they're recycling CO2 and maybe one other element I forgot. They're recycling it from the atmosphere and then using it for their needs, converting it into a organic form so that we could use it. Some of the archaea are extreme thermophiles, meaning they live in extreme temperatures, like 95 degrees Celsius or hotter. However, only some of them are found in extreme environments. We do find members of the domain archaea in all of the biomes on the planet. So wherever we look, we find the domain archaea. They are, in fact, play a significant role in, well, not role, but significant numbers in the soil and in the plankton of the ocean. that a significant number of the cells are from archaea. Any question about any of that? All right, does anybody know, is any member of the archaea pathogenic? Does any archaea cause human disease? It's a yes or no question, people. You got at least a 50% chance of getting it right.
No,
Thank you. Yes, meaning you are correct. That is, no members of the domain archaea are pathogenic to humans. No member of the domain archaea is pathogenic to humans. So we're not really going to study much about the domain archaea after this lesson, because our focus in this class is talking about infectious diseases and the microbes that cause them. And since none of the infectious diseases are caused by an archaea, this class is not really going to be studying the archaea. Any question about any of that? All right, let's talk about the domain eukarya. We did mention that this domain has 4 different kingdoms: kingdom Plantae, kingdom Animalia, kingdom Fungi, and then, excuse me, kingdom Protista. All of the cells of the domain eukarya lack peptidoglycan. So generally speaking, the only members of living organisms which contain peptidoglycan belong to the domain bacteria. That's the only domain that have peptidoglycan. Any question about any of that? All right, let's talk a little bit about the kingdom fungi. Fungi is plural, fungus is singular. You don't really need to know about what makes up the cell walls of fungi, but you should know that they use organic chemicals for their energy needs, like generally glucose. And that should be familiar to you because we use organic chemicals as our food source. Molds are multicellular, and then mushrooms are multicellular, but the molds tend to have a mass of mycelia, which we're looking at here. And then the mycelia are made up of individual cells that we call hypha. Yeasts, on the other hand, are not multicellular. They are singular. And some of the yeasts, most of the yeast are decomposers, but some of the yeasts are pathogens. Any question about that? For example, a pathogen yeast is Candida albicans, and that yeast can cause human disease. Somebody's got their microphone on. If you could turn it off, please. All right, let's move on to the kingdom Protista, the protists. As I mentioned, this kingdom is not monophyletic. It never was. It's simply defined as a eukaryote that is not a plant, not an animal, and not a fungus. Some of the kingdom Protista are algae, and they are plant-like protists. They have cellulose in their cell walls. They are photosynthetic, meaning they make their own food from light, carbon dioxide, and water. They do tend to produce molecular oxygen, as well as organic chemical compounds such as glucose. The Next grouping of the kingdom Protista is the protozoa, such as amoebas, parameciums, and euglena. These organisms absorb or ingest organic chemicals, such as an organic chemical can be here, and then this will close off, and that will be brought inside the cell. The kingdom protozoa, actually there's the kingdom Protista, and then the grouping of protozoa include the amoeba, the paramecium, and the euglena. They may be motile or non-motile. If they're motile, they can move by pseudopods, and there's one there. And that's where the cell, if it's moving this way, it pulls in the cytoplasm and the cell from this side, and then puts it out on this side. And that is what this organism moves by. And this is an amoeba or a white blood cell moving by pseudopods. So if it's moving this way, you put the cytoplasm down here and you pull it up in this region because the cell's moving that way. The protozoa can also move by cilia, and that will be a small hair-like object, and then all of them beat at the same time, these small hair-like, and that'll propel the cell forward. We'll talk a little bit more about that in a little bit. And then the third way that the protozoa can move is they can have a flagella, and that would be a whip-like structure. We talked about that with the prokaryotes. They also have a flagella. The difference is the eukaryotes can move by three ways: pseudopods, cilia, or flagella. The kingdom Archaebacteria or bacteria, and then the kingdom not able to say it. The prokaryotes only move by one way. They can only swim or move by one way, and that is by having a flagella or flagellum, more than one. More than one is flagella. Flagellum is singular. Sorry, got that reversed. But the eukaryotes can move in three different ways: by pseudopods, cilia, and flagella. Some of the protozoa are parasitic, and they can cause human disease. Like a tapeworm infestation would be a human disease caused by protozoa. And then the third group of the kingdom protists are the fungus-like protists, and they don't cause human disease, so we're not really going to talk much about the fungus-like protists. If you want one disease that's caused by the fungus-like protist is the potato rot. It has another name, which I'm escaping me what the heck this other name is. But potato blight, where the organism infects the potato, the plant, and then causes the tubers either not to grow or to rot. Any question about the kingdom Eukarya? Protozoa are the most important members of the kingdom Protista because the protozoa can cause human disease. Infectious human disease, I should add. All right, so looking at the kingdom Plantae, they are composed of mosses, ferns, conifers, and flowering plants. All of the members of the plants are multicellular. We're not going to study plants because they do not cause human disease. Of the kingdom Animalia, this includes the sponges, the worms, and the scientific name would be a helminth, insects, and the vertebrates. You can split the kingdom Animalia into more groups than this, but we're just going to leave it like that and say that there are a grouping of sponges, worms, the helminths, the insects, and the vertebrates. All of the animals are multicellular. And then one of the members of this group can cause infectious disease. And that is the helminths, the worms, can cause disease, an example being tapeworm infestation. All right, any question about any of that? So let's talk a little bit more about what comes under the umbrella of a microorganism. We already mentioned it does include the bacteria, the archaea, and I think I mentioned last time that it also includes viruses and prions. These members are special because they're not cellular. Virus and prions are not an organism. So generally, I'll call them a microbe. I'll try not to call them a microorganism because they are not organisms. But when we use the term microorganism, we don't necessarily mean that it is cellular. So if you're talking about like HIV, you can say it is a microorganism. I will try not to use that term just because I think it's a little confusing. It is, in one sense, a microorganism, but it is not an organism because it doesn't have cell. And so I'll just call it a microbe. Then there's the eukarya, the protozoa, which do cause human disease, and then the molds and the yeast, which also can cause disease, like Candida albicans can cause human disease, especially on the skin, but it can cause disease in other places, especially the mucous membranes. Some of the microbes of the domain eukarya can cause disease. Most do not. Most would be the algae. And then the helminths can cause disease. When we're talking about the microbes being a helminth, why are we saying a parasitic worm is a microorganism? Because most parasitic worms are not microscopic, right? Most parasitic worms are not microscopic. Nobody answered that, so I'll just answer right myself. Like the human tapeworm is something like 6 meters long, or can grow to that size. 6 meters long, essentially spanning one length of the intestine all the way across the intestine, meaning from the beginning of the intestine to nearly the end of the intestine. So why do we call the worms a microorganism if most worms are not microscopic? Nobody has a guess? The reason why we call them a microorganism is because microbiology studies things that cause infectious disease, and in some infectious diseases are caused by a helminth, a worm. Another reason why they should be called a microorganism is because generally it is a microscopic stage of the helminth which infects the human. You don't get an adult worm infecting a human. You have to have a microscopic stage of the worm infecting the human. Any question about any of that? All right, let's talk a little bit more about the helminths. They're multicellular animals. Many, but not all of them, are parasitic. Like all of the flatworms are parasitic. I think that's right. And many of the roundworms are parasitic. But not all worms are parasitic. For example, earthworms, none of them are parasitic. None of them cause human disease, an earthworm. You have to have a flatworm or a roundworm. And then many of the roundworms, probably most of the roundworms, are not parasitic. They do not cause human disease. And I can't remember the name of that roundworm, which is not parasitic. It's a free living roundworm, which is microscopic. And I forgot his name, so not a big deal. All right, any question about the helminths? One thing you should know about the helminths is that there are 3 groups of helminths, or 3 different worms. There's a earthworm, a flatworm, also called a platyhelminth, and then a roundworm called a nematode. And you can call them either a roundworm or a nematode, or a flatworm or a platyhelminth. And then the earthworm, which I think that's grouping Anilda, but I have to look that up, so don't quote me on that. And each of these groups are as different from each other as they are different from a human. So in a sense, we shouldn't lump the helminths all together because they are genetically as different from each other as they are from humans. Any question about any of that? If there's no questions, it's time to end this lesson. So I will see you at 6:30 at the lab. And by the way, between 6:15 and 6:20, if I keep talking, you can remind me that what the time is, and then we'll finish up. Because I went a little over today. All right, I'll see you at 6:30 in the lab.
Okay, thank you.
All right.